Our third speaker is Élyse Caron Beaudoin. And Élyse is a assistant professor at the University of Toronto at Scarborough. And she was the recipient of the 2025 ICEDA Early Career Award. And her research integrates toxicology, exposure science, epidemiology, and in community engaged approaches to better understand how environmental contaminants affect endocrine systems and community health. And her talk is going to be about endocrine disruption across scales and linking molecular mechanisms to community health. Élyse, I look forward to your presentation.
Perfect. Thank you so much for the introduction. Hopefully, you guys can see my screen correctly.
Yes.
Awesome. Thank you, Remy. Perfect. So thank you so much for the invitation. Really happy to be with you today. Before I start, I want to acknowledge the land on which the University of Toronto operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home of many Indigenous people from across Turtle Island, and I'm very grateful to have the opportunity to work, live, and play on this land. So today I'm going to talk about endocrine disruption across scales, linking molecular mechanisms to community health. And before I get started, I wanted to briefly go over my academic background to kind of give a sense of, I guess, why I do the work that I do. So I have a bachelor's degree in biology from the Université du Québec à Montréal, and then I did at the same university a master's degree where I used stable isotopes and GPS-based telemetry to understand the associations between diet and exposure to also brominated flame retardants like Madeleine is studying, but this time in ring-built galls. After my master, I did my PhD also at INRS, and during my PhD, I developed and used bioassays with molecular biology and genetic techniques in the context of endocrine disruption during pregnancy and during the development of breast cancer. And it really gave me a solid foundation to better understand complex interactions between our environment and human health. And then in 2016, Marc-André Werner at the Université de Montréal and myself instigated a pilot study on exposure to contaminants associated with unconventional oil and gas activity in northeastern British Columbia. And this led in 2017 to the start of my postdoctoral fellowship with Marc-André Werner on exploring the associations between proximity to this industry and birth outcomes. And then in 2020, I started my assistant professor position at the University of Toronto in environmental health. So at the University of Toronto, I lead the From Bench to Communities Lab, which is committed to integrating laboratory science with real-world community needs. So across these different stages of my career, a unifying theme has been understanding how environmentally relevant exposures to contaminants translate into biological disruption. And I started to really study this question during my PhD at INRS. For example, this first paper from my PhD shows that exposure to atrazine, which is a herbicide, and some neonicotinoid insecticides disrupt the expression of aromatase, which is a key enzyme in estrogen production, and they do that in a promoter-specific manner. Using H295R cells, which is a widely used human adrenocortical carcinoma cell line to study disruption of steroidogenesis, we also showed that these pesticides increase the biological activity of aromatase, which you can see on the right side of the figure here. In this next paper, we used a physiologically relevant fetoplacental co-culture model that was developed by Cathy Vaillancourt at INRS, and we used this model to assess how these neonicotinoid insecticides affect hormone production during pregnancy. And we showed that these pesticides disrupt estrogen synthesis by increasing the production of estradiol and estrone while also suppressing estriol production, likely through interference with fetal steroidogenic pathways involving the enzyme CYP3A7. As we can see on the figure on the right, when we supplemented the culture media with estriol, it reversed the neonicotinoid-induced increase in CYP3A7, and that supported kind of a feedback mechanism where reduced estriol levels is what drive CYP3A7 upregulation. And then finally, in this third paper of my PhD, we used a breast cancer cell line called HS578T. And we found that the expression of CYP19, so aromatase, via promoters, specifically P2, 1.3, and 1.7, was in part dependent on the activation of two VEGF signaling pathways. In this study, the exposure of our breast cancer cells to environmentally relevant concentrations of two neonicotinoids called imidacloprid and thiacloprid, it resulted in a switch in aromatase promoter usage involving the inhibition of 1.4 promoter activity and an increase in P2, 1.3, and 1.7 promoter mediated CYP19 expression. And what was interesting is that in this study, greater effects were seen at the lower concentrations that we used. So kind of to wrap up this, early kind of years in my career, our work, as with many others, including those presented today, we show that endocrine disruptors can reprogram hormone production at multiple levels, from gene expression to enzymatic activity. The work that I did during my PhD showed that environmentally relevant concentrations of various pesticides can alter key sterogenetic pathways in different models relevant for reproduction or cancer. And these effects were mediated by specific molecular mechanisms, and that straightened causal interpretation. So overall, this research highlights the need to integrate mechanistic and human relevant models to better assess endocrine disruption and risk. So towards the end of my PhD, I started working, as I mentioned, with Marc-André Werner on the potential health impacts of living in proximity to unconventional oil and gas activity. Canada is one of the main producers of unconventional natural gas in the world, with most of this industrial activity taking place in Alberta, as well as the northeastern part of British Columbia. To extract natural gas, the industry uses a technique called hydraulic fracturing. And the main phases of this whole process are first the pad preparation, where the site is prepared, then the drilling phase, where the wells are actually drilled vertically and then horizontally, as you can see on the image here, and where holes are punctured in the horizontal section of the well. The third phase is the hydraulic fracturing or stimulation phase. This is when large volumes of fracking fluids that contain water, sand, and various chemicals are injected in those wells, and it creates fractures in that horizontal part of the well, and that frees the trapped natural gas or oil. And then finally, the last phase is the production, where the natural gas or the oil will flow freely to from the into the well to the surface. So this is a kind of a map of the northeastern corner of British Columbia, and all the little triangles that are either yellow or green represent individual oil and gas wells. So in yellow, you have those unconventional oil and gas wells that use hydraulic fracturing. This region sits on the Montney Formation, which represents approximately 67% of British Columbia's production of natural gas. And we know from previous scientific studies that there is potential contamination of the environment associated with this industry, namely by some air pollutants like volatile organic compounds, as well as trace and radioactive elements that are naturally occurring in the rock formation, but that are being demobilized by this whole kind of hydraulic fracturing process. And right now in northeastern British Columbia, although you see a lot of little green triangles, which are conventional oil and gas wells. Most of the active wells are unconventional. When we started working in northeastern British Columbia, Élyse Elliott and colleagues just published in 2016 a systematic evaluation of around 1,000 chemicals identified in hydraulic fracturing fluids and they evaluated these chemicals for their potential reproductive and developmental toxicity. And what they found is that for 76% of the identified chemicals, information on reproductive and developmental toxicity was lacking. And for the remaining 24% of those chemicals that had some toxicological information, 43% of them were potential developmental toxicants, 40% of them were potential reproductive toxicants, and 17% of them were both. It's important to know that when this study was published now 10 years ago, there was no peer-reviewed exposure assessment studies in the context of unconventional oil and gas in the scientific literature. Now, there's about 54 epidemiological studies regarding the potential health effects associated with this industry, and a good proportion of those studies investigated birth and pregnancy outcomes, and most of those found significant associations between maternal proximity to unconventional oil and gas and negative outcomes such as preterm birth, low birth weight, and congenital malformations. So in 2016, when we looked at the available toxicological data, it did tell us that reproduction and fetal development might be affected by exposure to this industry. And so we were first interested in conducting an exposure assessment pilot study in pregnant individuals living in northeastern British Columbia. So we collaborated with medical clinics and First Nations communities in the region. And what we found at the time was that the median level, and we collected in those participants urine samples as well as hair samples, and we measured a suite of contaminants and then compared those levels with data in the general population. And what we found was that the level of, for example, one benzene metabolite, benzene is a volatile organic compound. One of its main metabolites is called trans-meconic acid. The level of TTMA was higher in our pilot study compared to the Canadian general population. And it was even higher when we specifically looked at the Indigenous participants in our pilot study. We also measured different trace elements or metals in the urine and hair samples. And when looking at the results in hair, we found that levels of elements like barium, aluminum, strontium, and manganese were also higher in our pilot study participants compared to reference populations. And for some of those elements, the concentrations were even higher in Indigenous participants compared to non-Indigenous participants. So this pilot study was the catalyst of two major studies that are ongoing and part of my research program. So when I started my postdoctoral fellowship with Marc-André Werner, I started working on these epidemiological studies. And so based on, at the time, what we knew in the epidemiological literature, the toxicological data regarding those contaminants, we hypothesized that proximity to unconventional oil and gas development in northeastern British Columbia could contribute to poorer birth outcomes. So we used birth records of approximately 6,000 births that happened between 2007 and 2016 at the Fort St. John Hospital, which is the largest hospital in the region. For each of these births, we had information about the mom, including the postal code, which was associated with the residence of the mom at the time of delivery. And so we use that postal code to estimate the activity, the oil and gas activity around, near the participant's home. And we calculated a series of metrics based on the density of wells around the participant's home or the postal code, using different buffer zones. We also calculated a series of metrics that consider both the density of wells, so how many wells are around the participant's home or postal code, as well as the distance of those wells to the residence or the central point of the postal code. And we accounted for all the wells in the province, and then we also use different buffer zones around the residence or the postal code. And then later on, a few years later, with now my PhD students, we were able to even refine more these proximity-based metrics by making the distinction between the types of wells, so are they conventional or unconventional, and in which phase of development they are. These metrics are called IDW, and they stand for inverse distance squared weighting approach. So in that first study, we evaluated the association between those IDW metrics and birth weight. And on the figure, you can see the adjusted beta for the multiple regression models with their 95% confidence interval. And you see for two buffer zones here, the 10 and the 5 kilometer, as well as for each quartile of exposure. So a beta is the average amount by which the birth weight would change when the IDW metric increased by one standard deviation, and if all the other covariables remain constant. So a beta of 0 would mean that there's no relationship at all between the IDW metric and the birth weight. So what we found is that after adjusting for covariables, we found that compared to women in the first quartile, women in the second quartile when we use the 10-kilometer buffer, and women in the second and third quartiles when we use the 5-kilometer buffer, had babies that were smaller than in the reference quartile. So the birth weights were between 41 and 47 gram lower than the first quartile, so the reference group. We also evaluated the association between metrics of residential oil and gas, so the same IDW metrics, and mental illness and substance use among pregnant women in northeastern British Columbia. And what we found is that in the second and third quartile of the IDW metrics, when we use the 10-kilometer buffer, we observe a positive association with depression compared to the first quartile. And when using a bit of a larger buffer zone, we found that the third and fourth quartiles of the IDW metrics were associated with greater odds also of depression, but anxiety and substance use during pregnancy compared to the first quartile. So the 2016 pilot study that I showed earlier allowed us to observe high levels of 1-benzene metabolites and several trace elements in urine and hair samples of pregnant people from northeastern British Columbia. And we know that these compounds can transfer through the placenta and reach the fetus. So in a larger biomonitoring study, we wanted to confirm or disconfirm the results from that initial pilot study. And also because endocrine disrupting activities have been observed for a variety of chemicals related to unconventional oil and gas in other studies, we wanted to investigate the endocrine disrupting potential of environmentally relevant concentrations of contaminants associated with this industry. So I'm going to present to you some results from that study called Exposures in the Peace River Valley, or EXPERIVA study. So for EXPERIVA, we recruited 85 pregnant people in the summer of 2019, and we worked with medical clinics, a midwifery clinic, as well as First Nation partners in the region. And we collected a lot of information in EXPERIVA. So for the exposure assessment, we used a multimedia approach combining environmental monitoring of VOCs and trace elements in tap water and in indoor air. And we also did biomonitoring of trace elements and VOC metabolites in urine, hair, and nails. We also measured a suite of oxidative stress and chronic stress biomarkers in the urine samples. And using biomonitoring data, we reproduce mixtures of trace elements to expose in vitro and ex vivo models to assess endocrine disrupting potential. And that last part is being led by Géraldine Delbais and Cathy Vaillancourt at INRS. And we also use the same IDW technique to assess the intensity of oil and gas activity around the participants' home during their pregnancy. So here, I just wanted to show you what it actually looks like in terms of intensity of activity. So here, this image is a zoomed in map where the green dots represent individual oil and gas wells. There's around 30,000 in the region. And the purple dots represent approximate location of the Xperva study participants' residence. So you can appreciate how close the wells can be to the residences. It's important to note, however, not all of these wells are necessarily active. Some may be abandoned or in different phases of production. In Xperva, the median number of wells around a participant's home in the buffer zone of 10 kilometer was 90 wells, and the maximum was 346 wells. So we measured different VOCs in indoor air, and you can see the levels of those VOCs in Xperva in the blue-green bars, and the median level of those same VOCs reported in the Canadian Health Measure Survey, or CHMS, in yellow and orange. So CHMS provides nationally representing levels of VOCs in indoor air. So for 27 out of the 40 VOCs that we measured in residential air, median levels in Xperva were between 1.3 and 3 times higher than the national median levels. And in particular for the VOCs that you now see in red boxes, the 95th percentile of the Canadian Health Measure Survey was exceeded by a substantial percentage of the Xperva participants. My PhD student, Corinne, then looked at the associations between these IDW metrics and indoor air VOC concentrations, and she found that the density of unconventional wells was associated with increased indoor air chloroform and D5. And when using the phase-specific metrics for unconventional wells, she also observed that the drilling phase was particularly associated with increased indoor air chloroform, and the production phase was associated with increased benzene. We also measured trace elements in urine and compared with general populations in Canada and the United States. And what we found is that the median urinary levels were higher in Xperva for barium, cobalt, strontium, and that the 95th percentile of reference populations was exceeded by a substantial proportion of the Xperva participants for barium, cobalt, copper, manganese, selenium, strontium, and vanadium. And for some of these contaminants, these elements, the concentrations were higher in indigenous participants. Unconventional oil and gas, as I explained, contributes to emissions of VOCs and trace elements, and some of these contaminants are known or suspected endocrine disruptors, and they can also promote oxidative stress. So my PhD student, Matthew, was interested in measuring a suite of oxidative stress and antioxidant markers in the urine samples of our participants and investigate the associations with those IDW metrics. We know that oxidative stress can be linked to negative birth outcomes, such as preterm birth. And so what Matthew found was that the density and proximity of all types of wells was associated with increased catalase, which is an antioxidant enzyme, as well as a decrease in 8-OHDG, which is a biomarker of DNA repair. That may be linked to defective DNA repair mechanism in response to this exposure. Matthew also found that the density and proximity of unconventional wells specifically was associated with a decrease in SOD, which is another antioxidant enzyme. And Corinne was interested in looking at stress hormones specifically. As we saw in our previous epidemiological study, the increased odds of negative mental health outcomes during pregnancy, such as depression and anxiety. So using the urine samples in Xperva, Corinne explored the associations between those IDW metrics and the concentrations of various stress hormones, such as cortisol. That's what you see here. So in her main adjusted models, she found that cortisol concentrations were significantly higher in the highest quartile of exposure to unconventional oil and gas wells, specifically during the drilling phase. Those are the numbers you see in red and in bolded black. And then she observed similar results in other models that were further adjusted for gestational week and self-reported mental health status. And then finally, this study is conducted by Rida Balbaki, who's a PhD candidate that I'm co-supervising with Géraldine Delbais and Cathy Vaillancourt at INRS. And Rida showed that mixtures of trace elements associated with unconventional oil and gas and measured in experiva, such as aluminum, manganese, barium, cobalt, and strontium, can directly stimulate testosterone production in rat fetal testes, even at concentrations that were relevant for human exposure. And the figure on the right here also demonstrates an increase in the expression of 3-beta HSD, which is a key steroidogenic enzyme, with mixture exposures producing similar or stronger effects than individual elements, highlighting a mixture-driven disruption of fetal steroidogenesis. So to conclude on that part, we have now, epidemiological and toxicological evidence that links proximity to unconventional oil and gas activity with adverse birth and maternal outcomes, which are consistent with endocrine disruption. We also showed that Xperva participants had elevated exposures to different contaminants such as VOCs and trace elements. Those are known or suspected endocrine disruptors. And we have preliminary evidence that shows that density and proximity to those wells can contribute or be one of the factors that contribute to residential levels of those contaminants. We also saw that unconventional oil and gas activity was associated with alterations in endocrine-related biomarkers, including oxidative stress pathways and cortisol regulation. And finally, the experimental findings demonstrate that real-world mixtures of trace elements can disrupt hormonal pathways in biological models, thanks to Hida's work. So with that, I want to acknowledge our funding for those various studies, as well as our partners, the Treaty 8 Tribal Association, the Saulteaux, and the West Moberly First Nations, as well as a very long list of names of everybody who has participated at some point in the pilot study, the epidemiological studies, and the Xperva study. And also, I thank you for your attention, and I'm happy to take any questions.
Thank you very much, Élyse. That was a very informative presentation.
Thank you.
I don't, Romy, are there any questions from the audience, or do you want to?
We have one question for now.
Perfect. Thank you. Could you give that question?
Yes. Question from Vicky. Élyse, do you hypothesize the reduced birth weights are due to maternal adverse effects or directly on fetus or both?
That's such a good question. I think, well, this is all hypothesized. We haven't measured that in any of our studies, but I think it would be safe to say both. So in terms of, I'll take the example of our pilot study on, or exploratory study on maternal mental health. So we saw, increased risk of depression, anxiety, substance use in association with exposure, greater exposure to this industry. And we know, that negative mental health outcomes can also lead to negative birth outcomes for the fetus and the baby themselves. So I think there's definitely, some impacts through the pregnant person. And then there may also be impacts directly on fetal development through, oxidative stress or endocrine disruption. A lot of these contaminants are known or suspected endocrine disruptors. They induce toxicity in different types of animal and cellular models. So I would assume that it can come from both sides.
Thank you. We have another question from Vicky.
Okay.
Are there any regulations regarding how close drilling sites can be to residents in Canada?
Yes, that's an excellent question. Once again, we are currently working actually on putting together a policy paper around those distances that are called setback distance. In northeastern British Columbia, if I remember correctly, the setback distance is 100 meters. So that's the minimum distance between any sort of building, whether it's a residence or a hospital or, a public building and a well. So they are very, very close. They can be very, very close to residences. And this setback distance wasn't based on any human health risk assessment because those were not available at the time that the setback distances were put in place.
Thank you.
I
We have two other
go.
questions here. We have two
Oh,
questions.
go ahead, please.
One question from Bernard. Do you have any data on the role of the father's exposure on progeny outcome?
That's such a great question, Dr. Robert. No, we do not. I'm looking at Geraldine Delbas for that. I'm not that I'm aware of. There is, though, a really cool study called Presto, which is conducted in the United States and in Canada. And it's kind of a internet-based study. It's online, but the goal of Presto is to recruit couples that are trying to conceive, and they are collecting urine samples from the mother, well, the woman in the couple, and also sperm samples from dads. And there is a portion of Presto that is specifically interested in looking at oil and gas, unconventional oil and gas activity, and those kind of sperm quality concentrations parameters. So I don't think they have published anything yet, but there's definitely Presto in the works. But other than this study, I'm not aware of anything that was specifically conducted with the fathers in regions of unconventional oil and gas.
Hmm.
And thank you so much. We have another question from Isabelle. Since some persons from First Nations are still hunting and fishing, do you know if they have seen impact on animal populations? Do you see relationship between eating traditional food and exposure?
Yes, definitely. That's a great question. Thank you, Isabel. So it's definitely a concern. We're hearing that from our First Nations partners that a lot of people are avoiding to consume traditional foods because they are worried about contamination. And that's really sad because traditional foods are a great nutrition source of nutrition, and they're more affordable than market food, especially in rural regions like that. So we currently are starting a new CIHR project grant this summer where we will address this question specifically. So we are sampling traditional food samples where we will measure trace elements in those foods. And then we are also building a cohort of 150, hopefully, Indigenous adults. And we will assess their consumption of traditional foods as well as their exposure to trace elements and try to understand the relationship between consumption of traditional foods and exposure to trace elements in that region. It's being co-led by Marc-André Werner, Julian Napoleon, who's a member of Saulteaux First Nation and a biologist, as well as myself. So hopefully I'll have a better answer for you in a few years.
Well, that's a very interesting talk. Um, my question was going to be exactly what Isabel asked, so I won't I won't pursue that further. Um, but I was taken back by the comment that you made that the current regulations only have a 100, if I understood it correctly, a 100-meter buffer.
Yeah,
That seems incredibly small.
yes,
Uh,
it is.
and wow. And is there any movement by the regulating or regulators to modify that in some way?
We're trying to advocate for that. So we're working with our First Nations partners. We also spoke with City Council there, the Dawson Creek City Council, a few months ago to advocate for that. So we're trying. That's why we're also working on that policy paper to really kind of um, advocate for at least, using proper data to establish those setback distances. But yes, it is not very large at all. There's wells all over this region. It's hard to visualize when you've never been, but it's just really part of the landscape. And those wells are really small. They don't look like much. So it's, when
Mhm.
they're not being actively fracked and when they're not actively producing, it's really just a little thing out of the ground. It's not big at all. It doesn't take a lot of space. So I think there's maybe also that.
Yeah, okay. Well, I wish you I wish you good luck on that advocacy
Thank you.
aspect
We'll need it.
for sure. Yeah. Well, thanks. Well, maybe we will move to
Yeah.
our final presentation.